Cross-sectional anatomy, the brain. There is a systematic approach to cross-sectional anatomy brain interpretation. Symmetry. Compare left and right side of the cranium. Midline. Look for midline shifts. Cross-sectional anatomy, review anatomical landmark for each side. Brain tissue, gray matter and white matter. Intercerebral lesions. CSF, or central cerebrospinal fluid, space. Ventricle, basal cistern, cortical sulci, fissure. Skull and soft tissue. Scalp swelling, fractures, sinuses, orbit. Subdural windows. Look for blood collection adjacent to the skull. Bone windows. Skull, orbit, and sinuses. Intracranial air. This presentation will include the cerebrum, gray and white matter organization, cerebral lobes, basal nuclei, diencephalon, lymphic system, brainstem, cerebellum, cerebral vascular system, and cranial nerves. The main sulcus that can be identified on CT and MRI of the brain is the central sulcus, which divides the precentral gyrus of the frontal lobe and postcentral gyrus of the parietal lobe. These gyri are important to identify because the precentral gyrus is considered the motor strip of the brain, and the postcentral gyrus is considered the sensory strip of the brain. Other gyri important for imaging includes the cingulate, parahippocampal, and superior temporal gyrus. We will cover this in the limbic system and temporal lobe. The two main fissures of the cerebrum are the longitudinal fissure and the lateral sylvia fissure. Located in the longitudinal fissure as the falx cerebri and super and inferior sagittal sinuses. The lateral fissure is a deep furrow that separates the frontal and parietal lobes from the temporal lobe. This is a sagittal T1 weighted MRI of cerebral lobes and central sulcus. This next image is a coronal T1 weighted MRI of the cerebral cortex and corpus callosum, and an axial T2 gray and white matter organization, mid-sagittal view of the corpus callosum. The cerebrum as a whole has many critical important functions, including thought, judgment, memory, and discrimination. The cerebrum consists of gray matter, neuron cell bodies, and white matter, myelinated axons. The cerebral cortex, the outermost portion of the cerebrum, is composed of gray matter, approximately 3 to 5 millimeters thick. The cortex not only receives sensory input, but also sends instructions to the muscles and glands for control of body movement and activity. Mid-sagittal T1 weighted MRI of corpus callosum and an axial T1 weighted MRI of corpus callosum. Deep in the cortex is the white matter, which contains fibers that create pathways for the transmission of nerve impulses to and from the cortex. The largest and densest bundle of white matter fibers within the cerebrum is the corpus callosum. This midline structure forms the roof of the lateral ventricles and connects the right and left cerebral hemispheres. The four parts of the corpus callosum from anterior to posterior are the rostrum, genu, body, and splenium. Here we have coronal T1 weighted MRI of cerebral cortex and corpus callosum, and an axial T2 weighted MRI of anterior and posterior commissures. Two other important bundles of white matter fibers are the anterior and posterior commissures. The cerebral cortex of each hemisphere can be divided into four individual lobes: frontal, parietal, occipital, and temporal. These four lobes correspond to locations in the cranium bones with the same name. Note how the central sulcus and the Sylvian fissure connect. Here's a label of the lobes of the brain. The frontal lobe is the most anterior lobe of the brain. The boundaries of the frontal lobe are the central sulcus, which separates it from the parietal lobe, and the lateral fissures, which separate it from the temporal lobe. The frontal lobe is also involved in speech production and contains the motor speech language center. Broca's area lies laterally on the inferior surface of the frontal lobe, dominant for language, typically in the left inferior frontal gyrus. The parietal lobe is located in the middle portion of each cerebral hemisphere, just posterior to the central sulcus. The horizontal portion of the lateral fissure separates the parietal lobes from the temporal lobe. The parietal lobe is associated with the perception of temperature, touch, pressure, vibrations, pain, and taste, and is involved in writing and in some aspects reading. The most posterior lobe, the occipital bone, is separated from the parietal lobe by the parietal occipital fissure. This lobe is involved in the consciousness, perception of visual stimuli. The primary visual area receives input from the optic tract via the optic radiation extends from the thalamus. The most posterior lobe, the occipital lobe, is separated from the parietal lobe by the parietal occipital fissure. This lobe is involved in the conscious perception of visual stimuli. The primary visual area received input from the optic tract. Here we have a sagittal T2 weighted MRI with basal nuclei. The temporal lobe is anterior to the occipital lobe and is separated from the parietal lobe by the lateral fissure. Conscious perceptions of auditory and olfactory stimuli and dominance for language are functions of the temporal lobe. Memory processing occurs via the amygdala and hippocampus. Clusters of gray matter located in the parahippocampus, campial gyri of the temporal lobe, located on the superior temporal gyrus is the auditory cortex, which can be divided into primary and secondary auditory areas. The primary auditory area, Heschei's gyrus, receives the major auditory sensory information from the bilateral cochlea, whereas the secondary auditory area, or Necky's area, is the center for comprehension and formulation of speech. Deep in the temporal lobe is another area of cortical gray matter termed the insula, or island of Riley, often referred to as the fifth lobe. Here we have an image, axial T1 weighted MRI of the basal nuclei. Another image, axial CT of the basal nuclei. We have a coronal T1 weighted MRI of basal nuclei. Ventricular system. A pair of crescent-shaped look like two bananas, are your lateral ventricles. And the third ventricle, basal ganglion, superior cerebellar cistern, are located medially and posteriorly to the fourth ventricles. So we look at the diencephalon, which is a complex of structures within the brain. The major components are the thalamus and hypothalamus. The diencephalon functions as a relay station for sensory information and as an interactive site between the central nervous system and the endocrine system. It is closely associated with the lymphic system. Here is a cross-sectional superior view of ventricles in relationship to the surface of the brain. Note the third ventricle is in the diencephalon. The two most superior cavities are the right and left ventricles. These ventricles lie deep within the cerebral hemisphere and are separated in the midline by a membrane known as septum palluteum. The lateral ventricles also consist of central portions called the body and three extensions, the frontal, occipital, and temporal horns. The junction of the body and the occipital and temporal horns form a triangular area termed the trigone. The lateral ventricles communicate inferiorly with the third ventricle via a pair of interventricular foramen called the foramen of Monro. The third ventricle is a very thin, slit-like structure, again located in the diencephalon, in the midline inferior to the lateral ventricles. The thalamus form the lateral walls. The third ventricle communicates with the fourth ventricle via a long, narrow passageway termed the cerebral aqueduct or aqueduct of Sylvius. The diencephalon is a complex of structures within the brain. Its major components are the thalamus and hypothalamus. This is a mid-sagittal T1 weighted MRI of the brainstem. The hypothalamus functions in integrating the activities of the autoimmune, autonomic, endocrine, and limbic system by helping to maintain homeostasis as it controls the regulation of temperature, appetite, sexual drive, and sleep patterns. In addition, the hypothalamus modulates the activity of the anterior and posterior lobes of the pituitary gland via the release of neurohormones. These hormones stimulate or inhibit the release of pituitary hormones. The pituitary gland is located in the sella turcica at the base of the brain. The protected location of this gland suggests its importance. It is sometimes called the master gland because it controls and regulates the functions of many other glands through the action of its 6 major types of hormones. The hypothalamus sends signal to the pituitary gland to stimulate or inhibit hormone production. Here we have a reconstruction coronal CT image identifying the pituitary gland and the surrounding vascularity. As previously mentioned, the thalamus make up a portion of the walls of the third ventricle and connects through the middle of the third ventricle by adhesions known as the massa intermedia. The thalamus serves as a relay station to and from the cerebral cortex for all sensory stimuli, with the exception of the olfactory nerve. The epithalamus is the most posterior portion of the diencephalon and comprises the posterior commissurate and pineal gland. The pineal gland, an endocrine structure, secretes the hormone melatonin, a serotonin-derived hormone that aids in the regulation of cardiac rhythms. As better said, circadian rhythms. Melatonin also helps regulate the production of hormones LH and FSH. The pineal gland sits on the roof of the midbrain, just posterior to the third ventricle and below the splenium of the corpus callosum. It is sometimes calcified, which aids in its detection on CT scans and lateral radiographs of cranium. The limbic system. The limbic system is a complex group of interconnected brain structures and fiber tracts located within and adjacent to the medial surface of the temporal lobes. These structures can contain crucial connecting pathways that extend to other areas deep within the midbrain, basal nuclei, and cerebral hemispheres. They have a common functional role in the emotional aspect of behavior. Particularly, the limbic system is involved in aggression, submissive, and sexual behavior, memory, learning, and general emotional responses. Structures of the limbic system includes the hippocampus, the amygdala, the olfactory tracts, fornix, eccentricate gyrus, and mammillary bodies. Hippocampus and amygdala. Here we have an axial T1 weighted MRI of the hippocampus and amygdala. Contained within the parohippocampal gyrus are the hippocampus and amygdala, prominent structures involved with the memory and emotion. The hippocampus is an important structure that has a strong role in the transition of short-term memory to long-term memory. The amygdala is an almond-shaped mass of gray matter located deep within the parahippocampal gyrus, anterior to the hippocampus. The amygdala coordinates the actions of the autonomic, and endocrine systems and is concerned with decision-making, emotional processing, and aggressive and sexual behavior. This is an axial CT of the hippocampus and amygdala. The olfactory tract runs underneath the frontal lobes and connects to the amygdala to bring information from the sense of smell to the limbic system. Here we have a coronal T2 weighted MRI demonstrating the fornix, cingulate gyrus, and hippocampus. The brainstem is a relatively small mass of tissues packed with motor and sensory nuclei, making it vital for normal brain function. Of the 12 cranial nerves, 10 originate from the nuclei located in the brainstem. Its major section or segment are the midbrain, pons, and medulla oblongata. Here we have coronal T1 weighted MRI of midbrain. The midbrain, or the mesencephalon, which is located above the pons at the junction of the middle and posterior cranial fossa, is the smallest portion of the brainstem. The midbrain is primarily composed of massive bundles of nerve fiber tracts. The midbrain surrounds the cerebral aqueduct, which contains CSF and connects the third and fourth ventricles. Here is an axial CT of the cerebral pinnacles and tectum. The pons is a large oval-shaped expansion of the brainstem, centrally located between the midbrain and the medulla oblongata. The pons create a prominent bulge as it lies just posterior to the clivus and anterior to the cerebellum. The term pons literally means bridge. This definition is appropriate because the pontine fibers relay signals between the spinal cord and the cerebral and cerebellar cortexes. Here we have an axial T1 weighted MRI of the pons and cerebellum. and a sagittal CT reformat of the pons. The medulla oblongata. Here we have an axial T2 weighted MRI of the medulla oblongata and olives. The medulla oblongata extends from the pons to the foramen magnum, where it continues as the spinal cord. The medulla oblongata contains all fiber tracts between the brain and the spinal cord, as well as vital centers that regulate internal activities of the body. These centers are involved in the control of heart rate, respiratory rate, rhythm, and blood pressure. This slide demonstrates a coronal T1 weighted MRI of the medulla oblongata and olives. The cerebellum. Here we have a sagittal T1 weighted MRI of the cerebellum. The cerebellum, which is referred to as the little brain, attaches posteriorly to the brainstem and occupies the posterior cranial fossa. The cerebellum is the coordination center for motor functions. Although the cerebellum does not initiate actual motor functions, it uses the brainstem to connect with the cerebrum to execute the variety of movements, including maintenance of muscle tone, posture, balance, and coordination of movement. The cerebellum consists of two cerebellar hemispheres. These hemispheres have an interesting appearance because the folds of gray matter resembles a cauliflower. Coronal image of a cerebellum. The vascular supply to the brain is unique. In comparison with the arteries of the body, the walls of the arteries in the brain are thin and weak, causing them to be susceptible to aneurysms and strokes. The veins of the brain do not contain valves. This lack of valves allows the blood to flow in either direction, creating a route for blood-borne pathogens to pass from the body to the head and vice versa. The capillaries of the brain are unlike those elsewhere in the body in that they do not allow movement of certain molecules from their vascular compartment into the surrounding brain tissue. This unique quality of impermeable is termed the blood-brain barrier, BBB. The brain receives arterial blood from two main pairs of blood vessels and their branches, the internal carotid arteries and the vertebral arteries, which make up the anterior and posterior circulation, respectively. The cerebral artery circle, or circle of Willis, is a crucially important among the four major arteries, the two vertebrals and the two internal carotids, feeding the brain. The circle of Willis is formed by the anterior and posterior cerebral, anterior and posterior communicating, and the internal carotid arteries. The circle of Willis is located mainly in the suprasellar cistern at the base of the brain. Many normal variations of this circle may occur in individuals. The circle of Willis functions as a mean of collateral blood flow between the cerebral hemispheres in the event of a block. Here we have a CTA of the circle of Willis. The venous system of the brain and its coverings are primarily composed of the dural sinuses, superficial cortical veins, and deep veins of the cerebrum. There are 12 cranial nerves, numbered from anterior to posterior according to their attachment to the brain. All but the first and second cranial nerve arise from the brainstem. Creating mnemonics to learn anatomy is a best practice. The mnemonic for cranial nerves are, oh, once one takes the anatomy final, very good vacations are heavenly. This is the end of our presentation.